So a very warm welcome to all the YouTube subscriber of our Futuromics Bioinformatics Made Easy. So today we are going to have a video on the topic survival analysis in R language. So as we all know that the language such as the Python or R is playing a very significant role with respect to the biological data analysis, and majorly once we are dealing with the bioinformatics data. So today we are going to have an tutorial where we are going to discuss about how using the RStudio environment, we are going to perform the survival analysis of the cancer data set by using the scripts which I'm going to share today itself, right? So as you all can see that you have to launch your RStudio on your Windows operating system if you have. And then once you want to know how the installations has to done for the RStudio as well as R, then you all of you can refer to my video, which I have given a step-by-step tutorial about the installation process for the R and also for RStudio. Okay, so now you can see that the first script or the command what you have to do give in this particular script is that you have to scratch the library, or else you have to pull the library from by dplyr. And then the most important library what we need today for the survival analysis is the survival Miller plot and the survival plot. So you all of you can search the name of the particular packages by going in the specific tool and install package. There are the two way, okay, how you can install the particular packages using RStudio using the CRN. You can just write the name of the package and then you can proceed for the installation, or else simply you can just write the command which I have given over here. So the two libraries we have installed for the survival Miller and the survival. And then we are going to run the script now in front of you. All of you can see that. Once we have done the installation about the library, so you all of you can see that the installation has proceeded. And then the next library we are going to install is survival. Okay, so then we are going to pull one specific data sets, that is lung. So if you want to pull any kind of data sets, just you have to write the command in the form of question mark and the lung. So if you just run the script, now you all of you can see that you will get the information over here that what basically this data is about. But if you want to know more about the data, then you can just write head lung. So basically once you write the command, it will give you the head information, is that what the data is about. Then if you want to know what is the class of the data, is nothing but what exactly it contains. It is one of the data frame, and the dimension of the data is a total data set, which is about 228, and 10 columns are there. So if you want to know what exactly the data is about, just you can write the command like view and in bracket lung. Once you just run this, now you all of you can see guys that we got the data over here. Just to understand more, now you can see that the total number of columns are 10. And you will see the overall data sets is given over here. You can make filters to just fetch the data accordingly with the columns if you're interested, or else you can simply just move ahead just to perform some analysis on that. Okay, so you all of you will get this script based on the analysis. And now we are going to pull the certain library over here based on the lung data set. Now we are going to move ahead to just get to know the two important columns over here, that is survival. And then we are assigning the data that is survival based on the time and the status. And then the class of the data, that is survival. And this totally we are going to just run the head. Okay, then finally we are going to just make some small visualization so that we can just come up with respect to some interpretation. So fit a survival curve. So you have to write the command like a survival fit, and then the columns you have to just mention, that is a time and status. What is your target column? So now I'm going to run the script with respect to the two target column, that is the survival fit with respect to the time and the status. And the data is automatically the lung data set. So we are going to just mention that fit the data based on the survival, right? So once I just run this, all of you can see that we are just making the data in the proper process manner. Then overall summary of the data you will find once you just run the script. So basically what we are doing over here, we are trying to fit the data based on the time and the status. And then the gender is another one target which we are interested to get to know that what is the survival ratio based on the time and the status of the patient and how the gender is affecting, right? And then once you just write the command like summary and as fit what we have done, then it is making the process based on the analysis. And finally, we are going to see overall data in the range based on the time. So now we are going to make some visualization. So basically we have a Kaplan-Miller plot. So for that we need to fetch the particular analysis based on the survivor Miller, what we have installed the package. And then we are going to run based on the two columns, that is time and status, based against the gender, and plot the fit. So now you can see basically all of you over here. So this is what the visualizations will be represent. But it is, as you all can see, that it is not very much easy to interpret. Okay, so we have to make some small changes over here. So for that, we have to call the library, that is survival Miller. And then we need to take help of GG survival plot. As you all know that GG plot is helping us to give some more colorful images. So that's why we are going to pull the library of GG survival plot, against which data sets is this nothing but the SFIT, what we have done the processing. And then finally, as you all of you can see that we got the output. But basically, you can just add some more colors based on the target data sets, as you all of you can see here. So this GG survival plot with SFIT, and then the data is based on the definitely we have selected the data, and the p-value, if we are interested to get the p-value, just we have to write true. And then the label based on the gender, like male and female, and then you can even give the title, right? And then the color representation based on the title, that is Kaplan-Miller survival curve for the lung cancer survival. So likewise, you can give the more interactive way to get the graph. But for a while, if you can see that it is good image, okay. But to give some additional information based on the legends title, okay. So for that purpose, you can all of you can add one more command over here using this script. And finally, all of you can see that the output is get updated, and now all of you can see that it is much better. Okay, so using this particular plot, we can even represent our data. For example, if you are using the TCGA data set, okay, based on the lung cancer or any other cancer type, so you can pull the data set. That is the next video I'm going to probably going to upload through my channel. So kindly stay updated. Okay. So as you all can see that this is basically what we have input our script. We are getting the survival probability. We are getting the p-value based on the gender as well as the title for the same, right? So I think this, all of you can use this image for representing in for your project context, or else any kind of conference, or even for the good piece of paper publication, right? So so I hope that all of you understand the flow. Definitely I'm going to share the script so that all of you can refer and then you can follow accordingly. Thank you all of you for having an overview. So stay tuned for the next video, and kindly subscribe to the channel so that you will get the least video with respect to the bioinformatics tutorials and the different new informations which we are going to upload through our channel. So thank you all of you.